We were excited by our data that suggested that the CCL21 vault might someday be used as a cancer therapy. One big advantage to this approach was that vaults appeared to be taken up very robustly by phagocytic cells. However, most cells are not phagocytes. We realized that if we wanted vaults to be used to deliver payloads to any cell, then we would need to make vaults targetable. So, what if we could just attach a zip code to a vault? 91244, heart. 35737, spleen. 66320, lung. 14181, prostate. Hi, I'm Lenny Rome, the Vault Guy. I'm a cell biologist and a nanotechnologist. My passion is a common particle of the human cell, called a vault. With the expert assistance of my daughter-in-law and budding superstar actress Tara, I'm trying to explain what vaults are to non-scientists. In my last video, I described how we developed the CCL21 vault as a potential new cancer immune therapeutic. Today, I want to discuss vault targeting and go over three things. First, I'll review a basic process by which cells take up materials called receptor-mediated endocytosis. Second, I'll describe a strategy we used to attach a tag on the end of a vault. And finally, I'll explain how we used the tagged vaults to get them taken up by a cancer cell. Stick around. There might be something new going up on the wall back there. A major challenge in drug development is the lack of suitable methods to deliver therapeutic agents, drugs, proteins, or nucleic acids to specific host tissues. I showed you in episode 16 how vaults were taken up by phagocytic cells, but these cells are designed to eat particles. We wanted to develop a way to target the vault to any cell or tissue, especially cancer cells. Active targeting of a protein or a nanoparticle can be accomplished by attaching antibodies, peptides, or some small molecule that can bind to cells. This binding is usually directed towards cell surface receptors. Human cells have receptors on their surface that bind to certain molecules, collectively called ligands. Often, the binding of the ligand to the receptor results in uptake of the ligand into the cell. This process is called receptor-mediated endocytosis, an elegant mechanism for internalizing molecules important for cell function. Let's describe the process using a hormone called epidermal growth factor, abbreviated EGF. EGF is an important cell regulator that has many functions, including stimulation of cell growth. EGF binds to a cell receptor, the EGF receptor, EGFR for short. EGFR is a transmembrane protein with a domain facing the outside of a cell, a domain which crosses the membrane, and a domain inside the cell in the cytoplasm. When EGF binds to EGFR, the binding alters the conformation or shape of the receptor, and this change sends a signal inside the cell. In addition, the binding of EGF triggers receptor-mediated endocytosis of the EGF-EGFR complex. The membrane invaginates and becomes coated with a protein called clathrin. This area is commonly called a coated pit. The pit deepens into the cell, and the membrane surrounds the receptor and pinches off into an internal vesicle called a coated vesicle. These are the very same vesicles I described in episodes 2 and 3 that we happened to be studying when Nancy Kadersha and I stumbled upon vaults. Antibodies can be produced that bind very tightly to cell surface receptors. In fact, antibodies against EGFR have been made, and these block EGFR signaling and that blocks the proliferation of cancer cells. We have used both hormones and antibodies as tools to target vault particles to cells. In episode 10, I told you how we mapped the position of MVP in the vault, and I described what happened when we added extra amino acids to the C-terminus of MVP. These extra amino acids ended up being displayed on the outside of the vault, at the top and bottom of the vault. One interesting peptide that we attach to MVP is called the Z domain. This is a 33 amino acid sequence that binds to antibodies, particularly antibodies of the IgG subtype. IgG is the most abundant antibody in blood. It is a Y-shaped protein composed of 4 polypeptide chains, 2 identical heavy chains, and 2 identical light chains. A molecular model of an IgG shows that the 4 chains wind around each other. Antibodies, especially IgG, play a critical role in protecting the body against bacteria and viral infections by binding to pathogens and neutralizing toxins and supporting immune cell function. Antibodies show high affinity, meaning very strong binding, to pathogens at specific locations on their surfaces, called antigen binding sites. There is also an important area called the Fc region, where immune cells and proteins of the blood clotting system can bind, and this is where the Z domain binds. The Z vault we made binds to an IgG like this at the top and bottom of the vault, but it does not block the antigen binding sites from binding to antigens. We wanted to know if the Z vault could target vaults to cells, so we used dendritic cells, DCs, as they have Fc receptors on their surface that bind to antibodies and play important roles in antigen recognition and uptake. DCs are phagocytic cells, and in episode 16, I explained how these cells just love eating vaults. Would they love vaults even more if we decorated the top and bottom of a vault with an antibody? We used Z vaults packaged with a green fluorescent protein inside, then examined DC uptake of the Z vaults in 30 minutes, both with and without decoration with an antibody. We found that 36% of the undecorated Z vaults were taken up in 30 minutes. However, uptake of the antibody-decorated Z vaults was 92%, indicating that targeting to the Fc receptors was working. In addition, we wanted to see if we could target this vault to non-phagocytic cells, so we used a cancer cell line that was known to overexpress EGF receptors on its cell surface. We decorated the green fluorescent Z vaults with an antibody that was specific for the EGF receptor. We then added these vaults to cells that express the EGFR on their surface and kept the cells on ice, which doesn't stop the vaults from binding to the cells, but does stop the cells from internalizing the vaults. The Z vaults decorated with the anti-EGFR were able to efficiently bind to the cancer cells. As a control, we used Z vaults that were not pre-bound to the antibody. When we added undecorated MVP Z vaults to the cancer cells, we saw no binding. We did one more interesting variation on the targeting. Instead of the Z domain, we expressed vaults with the 55 amino acids encoding the EGF hormone itself to the MVP C-terminus. Amazingly, these vaults retained the biological activity of EGF. They were able to bind to EGF receptors and induce receptor signaling, and even stimulate cell proliferation. I'm Lenny Rome, the Vault Guy. Join me in my next episode where we tackle the second big cargo delivery problem. That's getting vaults into the cell cytoplasm. As always, like and subscribe, and please comment on my lessons. Oh, one more thing. I've been waiting for an opportunity to hang 2 more pictures on my wall back there. One is a journal cover of a vault where we packaged a large lipid domain inside. This work was a proof of concept, showing that we could package lipid-soluble drugs inside the vault. The other is a terrific science article by John Travis that came out last year featuring vault research. I mentioned this article in my Welcome to the Channel video. Since it was the nicest thing that anyone has ever written about vaults and me, I wanted to get it up there on the wall as well. Well, you know the drill. See you in the next episode.